There is a word built from two other words, assembled in a laboratory in the 1970s by scientists who needed a name for something they had just found inside the brain. The first half of the word is endogenous, which means produced within the body. The second half is morphine, the most powerful painkiller extracted from the opium poppy. Put them together and you get endorphin, the body's own morphine. The scientists did not choose that name carelessly. They chose it because what they found in the brain was not merely similar to morphine. It fit the same receptors. It activated the same pathways. It had been doing this, unobserved, for the entire length of human history. The brain had been running a pharmaceutical operation long before anyone pressed the first poppy into a slab of clay. You've probably heard endorphins mentioned in connection with exercise. The runner's high, people say. The lift you feel at the end of a long run, or after an hour of climbing stairs, or after pushing through something that seemed impossible at the halfway point. You were told it was endorphins flooding the brain. That explanation is so widely repeated that most people accept it without pausing, which is why it matters that the physiology does not actually support it. The endorphins released in large quantities during exercise are produced by a gland, and they travel through the bloodstream. But the brain is not open to the bloodstream the way the rest of the body is. There is a border, and it is strict. Endorphins, as molecules, are too large and too water-soluble to cross it. The brain the runner's high story assumes they are flooding is, in the most literal sense, not receiving them. This is not a minor technical correction. It is the first clue that endorphins are doing something different from what the popular account describes. And once you follow that clue, you end up somewhere much stranger and more significant than a pleasant run. You end up at pain, at emergency analgesia, at expectation being powerful enough to activate the brain's own pharmaceutical system, at the neuroscience of laughter, of heartbreak, of why social rejection produces a sensation that is not merely painful in a metaphorical sense, but activates the same circuits as a physical wound. The story of endorphins is the story of a system the brain built to survive, not to feel good, to survive. The pleasure when it comes is almost incidental, and the fact that a leaf from a poppy plant can hijack that system so completely is not a curiosity. It is a window into something the brain has been carrying since long before it became a brain capable of wondering what a poppy is. So why does the brain have a receptor that morphine fits? And why does blocking those same receptors change not only physical pain, but the way a person feels when they are left out of a room? If you enjoy exploring questions like these, about what the brain is actually doing beneath the story you were told, a like would be highly appreciated. And if something raises a question or a thought while you listen, drop it in the comments. Now, let's begin. In 1972, a graduate student at Johns Hopkins University decided to look for something that every pharmacologist at the time assumed did not exist. Her name was Candice Pert. She had broken her back in a riding accident before starting graduate school and spent weeks on morphine during her recovery. The experience left her with a question she could not set aside. Morphine came from a plant. It had no obvious reason to work on a mammalian brain. Yet it worked with a precision and a reliability that seemed to demand an explanation. The drug did not merely blur things, it targeted something, it did something specific. She began to wonder whether the brain might have a receptor, a dedicated molecular binding site that opiates fit because something else had shaped it long before opiates arrived. Her supervisor at the time thought the idea was not worth pursuing. The conventional view held that drugs like morphine simply dissolved into cell membranes and produced their effects non-specifically, the way alcohol affects tissue. There was no reason to suppose the brain had evolved a dedicated architecture for a plant extract. Pert proceeded anyway. She developed an experiment using radioactive morphine, a tracer that would light up wherever the drug physically attached to tissue. The logic was simple. If there were a discrete receptor, The radioactive tag would concentrate there. If the drug simply dissolved everywhere, the signal would be diffuse and uniform. She ran the experiment during a week when her babysitter cancelled. Her 5-year-old son sat on the lab bench while she worked. The results came back showing concentration. The radioactive morphine was not spreading evenly through tissue. It was binding in specific locations, with a precision that looked nothing like a molecule dissolving into membranes. The brain had binding sites. There was something else in the data. The binding was saturable. You could add more and more radioactive morphine, and at a certain point the concentration in the specific regions stopped increasing, as if the sites had been filled and there was no more room. This is the hallmark of a real receptor. A non-specific membrane effect does not saturate. It simply keeps growing as more drug is added. Saturation means something specific is being occupied, something with a finite number of places, something that was built with a capacity. The brain's opioid receptor had a capacity, which meant it had a design. The paper was published in Science in 1973. 3 groups published independently that same year, all arriving at the same finding by different routes. Lars Torenius in Uppsala, Sweden, Eric Simon at New York University, and Pert and Snyder at Johns Hopkins. 3 laboratories on 2 continents, and all 3 found the same thing. The brain contains specific protein structures that bind opiates with high affinity. The receptor was real. It was embedded in cell membranes, wound into a shape that opiates fit with high specificity, and it was distributed through the brain in a pattern that was not random. It appeared in regions involved in pain, in emotion, in breathing. The distribution itself was a clue to what the receptor was doing. The mapping of where these receptors concentrated told researchers something important. They were dense in the dorsal horn of the spinal cord, where incoming pain signals are first processed. They were present in the brainstem regions that control breathing. They appeared in the limbic system, in the regions that handle emotional memory and the sense of threat or safety. This was not a single receptor doing one thing. This was a system embedded across the architecture of a nervous system built long before anyone pressed a poppy into medicine. 3 subtypes of the receptor were eventually characterized. The mu receptor is the most studied. It is the one morphine primarily activates, concentrated in the regions that gate pain and in those that process the pull toward things. The kappa receptor, found in different concentrations, is associated with a different profile of altered experience. The delta receptor has its own distribution and functions that researchers are still working out. Together, this family of receptors forms a coherent system, not a random scatter of binding sites. The architecture looked, even in 1973, like something that had been built to serve a purpose. And that was the problem the discovery created. The receptor existed. It was specific. It was distributed in a pattern that suggested an evolved function. But what was that function? The receptor had not evolved so that morphine would have somewhere to bind. Evolution does not work in anticipation of plant alkaloids. The receptor existed because the brain was using it for something internal, something that fit the same binding site and activated the same cascade of effects, which meant there had to be a molecule, something produced inside the body, something that reached these receptors under conditions that made physiological sense, that activated them naturally, that had been activating them for millions of years before anyone discovered the receptor at all. When the 1978 Albert Lasker Award was given for the discovery of the opioid receptor, Solomon Snyder received it. Candice Pert, whose experiment had been the central demonstration, was not included. She wrote a letter to the Lasker Committee describing what she regarded as an injustice, and continued publishing work on receptor biology for decades afterward. The award went to 3 men. The woman who had designed and run the key experiment spent years explaining what her role had been. This is not a digression. It matters because it reflects how clearly the discovery was understood, even at the time, to be significant. Lasker Awards are sometimes called the American Nobel. The committee does not give them for routine findings. The opioid receptor was, in the estimation of those who study the brain, a discovery of the first order. That question remained open for 2 more years. Then a pharmacologist in Aberdeen, Scotland, went looking for the molecule these receptors were built for. He found it in a slaughterhouse. Hans Kosterlitz was a pharmacologist in his early 70s when the opioid receptor was discovered in 1973. He had spent decades studying the way gut tissue responds to morphine, developing precise laboratory assays that could measure opiate activity with exceptional sensitivity. His method was indirect: strips of guinea pig intestine and mouse tissue that would contract or relax in response to opiates, giving him a biological readout of how powerfully any given compound activated the system. It was slow, meticulous work. It was also exactly the kind of tool someone would need to search for an unknown molecule in brain tissue. John Hughes was a pharmacologist and lecturer at Aberdeen University, and he was the one who went looking. In the early 1970s, Hughes began using Kosterlitz's assays to test whether extracts from brain tissue contained any compound that behaved like an opiate. The principle was straightforward. If the brain made its own opioid, it would show up in tissue from the brain when tested against Kosterlitz's gut strips. If no such molecule existed, the extracts would have no effect. The challenge was the brain itself. To extract enough material to test required large quantities of tissue, and large quantities of tissue from the brain required large quantities of brains. Hughes solved this problem with a bicycle and a standing arrangement. Every day he rode his bicycle to a local slaughterhouse near Aberdeen. He traded bottles of whiskey to the butchers in exchange for pig heads. He brought the heads back to the lab, processed the brain material using acetone, and ran each fraction through Kosterlitz's assays, month after month, testing fraction after fraction, looking for the compound that would activate the system. The assay could not tell him what the compound was. It could only tell him whether something in the current fraction was doing anything at all. A piece of gut tissue that relaxed when it should have contracted. A signal where before there had been nothing. The activity had to be confirmed before there was any point asking what was producing it. And when the activity appeared, it was buried inside a mixture of thousands of proteins and peptides. Getting from a signal to a structure meant separating fractions and testing them one by one. The inactive ones were discarded. What remained active was concentrated and tested again. The process had a discipline of its own. Each round brought the unknown molecule closer to isolation without yet being able to name it. He found 2 of them. In 1975, Hughes and Kosterlitz published the structures of 2 small peptides that activated opioid receptors with the same specificity as morphine. Each was 5 amino acids long. They named them met-enkephalin and leu-enkephalin, from the Greek word for brain. These were the molecules the receptors had been waiting for. The brain had produced them all along. Short chains of amino acids that fit the mu and delta receptors with a precision that made clear these receptors had been built for exactly this purpose. The brain had not borrowed the morphine receptor from pharmacology. Pharmacology had borrowed it from the brain. When researchers mapped where these peptides were concentrated in the nervous system, the pattern was not random. Enkephalins were found in highest density in areas long associated with pain processing and emotional response. The dorsal horn of the spinal cord, where sensory signals arrive from the body, limbic structures involved in fear and stress, brainstem circuits that govern basic survival responses. The distribution matched what the receptor locations had already implied. These molecules were not background chemistry spread evenly through the brain. They were positioned at specific junctures in specific circuits, and the circuits they occupied were the ones the brain had built to process suffering and threat. The receptor had pointed to a system. The molecules confirmed it was real. What remained was to understand how it worked. The story did not end there. The enkephalins Hughes and Kosterlitz found were small molecules, 5 amino acids each. But researchers at other institutions were finding evidence of larger opioid peptides. Working with pituitary tissue from camels, Cho Hao Li and David Chung identified a much larger molecule, 31 amino acids long and more potent than morphine. They published the structure in 1976. This one was named beta-endorphin, from the portmanteau of endogenous and morphine. Its greater potency relative to morphine was confirmed the following year. Both the enkephalins and beta-endorphin came from precursor proteins. The body does not synthesize small opioid peptides directly. It builds a large protein first, then cuts it into functional fragments using specialized enzymes. Beta-endorphin comes from a precursor protein called pro-opiomelanocortin, or POMC. This same protein is also cut to produce a hormone involved in stress response, and another involved in skin pigmentation. The body uses one master protein to generate several distinct signals, each suited to a different tissue and a different purpose. The architecture of POMC reveals something about where beta-endorphin sits within the body's larger economy. It is not a comfort signal that the body added separately, somewhere alongside its more urgent systems. It shares its molecular origin with the hormones deployed when the body perceives a threat. The same enzymatic cut that produces a stress response hormone can produce an internal opioid, depending on which tissue is doing the cutting and which enzymes are active. Pain modulation and threat response are not separate systems that happen to interact. At the level of the precursor protein, they are produced from the same source. The endorphin system was never a luxury. It was built into the machinery the body uses to survive. The enkephalins come from a different precursor entirely. So do the dynorphins, a third family of opioid peptides identified shortly afterward. 3 families, 3 precursor proteins, 3 sets of opioid peptides, each with its own distribution in the brain and body, each binding opioid receptors with somewhat different affinities and producing somewhat different effects. The question was no longer whether the brain made its own opioids. The question was what they were for. Hughes and Kosterlitz shared the Albert Lasker Award in 1978, alongside Solomon Snyder. Hughes later became a Fellow of the Royal Society. Kosterlitz, who was already past the typical retirement age when the most important findings of his career were published, continued working at Aberdeen until well into his 80s. The scientist who had refined the assay over decades, and the younger pharmacologist who had ridden his bicycle to the slaughterhouse every morning had found, in pig brains and guinea pig gut strips, one of the most significant molecular systems in the history of medicine. Beta-endorphin was produced in 2 places. One was the pituitary gland, the small structure suspended beneath the hypothalamus that serves as the brain's main interface with the body's hormonal system. The other was a cluster of neurons inside the hypothalamus itself, specifically in the arcuate nucleus, a region deep within the medial portion of the hypothalamus. Both produced the same precursor protein. Both cleaved that protein using the same enzymatic process. But what they did with the resulting beta-endorphin was entirely different, because the 2 locations had entirely different relationships with the rest of the body. The pituitary is an endocrine gland. Its purpose is to release hormones into the bloodstream, where they circulate and act on distant targets. When the pituitary cleaves its POMC protein, the products go into circulation alongside stress hormones. Beta-endorphin is released during stress, injury, and sustained physical effort. It appears in the blood at measurable concentrations. This is the release that generates most of the popular discussion about endorphins. It is measurable. It is real. Researchers can detect it with blood samples, correlate it with exertion or pain, and watch its concentration rise and fall. The release is genuinely happening. What the popular account gets wrong is not whether it happens, but what it reaches afterward. Between the bloodstream and the brain lies the blood-brain barrier. It is maintained by specialized cells that line the capillaries running through brain tissue, packing together with unusual tightness so that the gaps between them are effectively sealed. What crosses must either be lipid-soluble enough to dissolve through the fatty membranes of those cells directly, or small enough to fit through specific transport channels the cells actively maintain. Alcohol crosses the barrier, THC crosses it, heroin crosses it, and does so rapidly, which is part of what makes it pharmacologically potent. These molecules can cross because they are lipid-soluble. They dissolve into the fatty membrane of the capillary wall and diffuse out the other side. Beta-endorphin cannot do this. It is a peptide, water-soluble, and 31 amino acids long, with a structure that dissolves in blood but not in membranes. There is no transporter waiting for it. The barrier does not recognize it as something to carry across. Beta-endorphin remains in the blood, and the brain it is supposedly flooding is not receiving it. The barrier's role was not merely inferred from molecular properties, it was tested directly. Injecting beta-endorphin into the bloodstream of animals produced detectable changes in peripheral pain responses, but did not produce the central effects of opiates. Injecting the same molecule directly into the brain produced potent analgesia. The barrier's exclusion was confirmed not by theory, but by the different outcomes from two routes of delivery. This does not mean the circulating beta-endorphin is doing nothing. Peripheral tissues throughout the body carry opioid receptors, and beta-endorphin in the blood can activate them. Nerve endings in inflamed tissue have opioid receptors that respond to opioid peptides in their local environment. Immune cells carry opioid receptors as well, and in some cases release their own enkephalins at sites of injury, contributing to local pain modulation entirely outside the brain. The peripheral system has a genuine role in reducing pain at the level of the body's tissues, but it cannot generate the brain states people are describing because it cannot reach the brain. The second system does not face this problem. The neurons of the arcuate nucleus release beta-endorphin within the brain, at synapses, acting on the opioid receptors of other neurons. Their axons project into multiple regions of the hypothalamus and extend down into brainstem nuclei involved in coordinating pain suppression. The beta-endorphin these neurons release never enters the bloodstream. It is synthesized inside the brain, released inside the brain, and binds to receptors inside the brain. It does not need to cross any barrier because it was never on the wrong side of one. And unlike the enkephalins, which act within microseconds and degrade almost immediately, the beta-endorphin released from these arcuate neurons can produce longer-lasting changes in opioid tone within the circuits it reaches. The system operates more slowly than the rapid local signaling of enkephalins, but it reaches farther within the brain and sustains its effects longer. The enkephalins, for their part, are the brain's nearest approach to a point-of-action signal. They are produced by neurons within the spinal cord and brain and released locally at synapses into the immediate space around them. They are broken down by enzymes within seconds of release. The molecule does not travel to find its target. The molecule and its target are already in the same location, and the action is complete before the molecule has moved any distance at all. Enkephalins are not a systemic signal. They are a precise local event. The fact that the same precursor protein generates beta-endorphin in both the pituitary and the arcuate nucleus reflects something important about the POMC system. The protein itself does not determine function. The enzymes that cleave it, and the tissue in which the cleavage happens, determine what gets produced and where it goes. In the pituitary, POMC is cleaved to produce beta-endorphin alongside ACTH, the hormone that triggers cortisol release from the adrenal glands. These products go into the blood together as part of the body's coordinated stress response. In the arcuate nucleus neurons, the same protein is cleaved to release beta-endorphin into local circuits. Same molecule, same origin, entirely different deployment depending on which tissue holds the scissors. When people describe the warmth of a genuine social moment or the altered state at the end of a long run, they are describing something that originates in the brain. Those experiences involve opioid receptors and opioid peptides, but the source is not the pituitary contributions cycling through the blood. It is the internal system, the arcuate nucleus neurons and the enkephalin-producing neurons within the brain circuits, releasing opioid chemistry locally in response to whatever those circuits are registering. The popular account names the right molecule and describes real experiences. The mechanism it implies is the one part it gets wrong. What those circuits are actually doing, and how they suppress a pain signal before it reaches conscious awareness, is where the mechanism becomes clearest. In 1969, a neuroscientist named David Reynolds reported that he had performed surgery on rats using nothing but electrical stimulation of a specific brain region as the anesthetic. The rats were awake. The procedure was otherwise identical to any standard operation, and the animals showed no pain response. Reynolds had located a region of the brain that, when stimulated, produced analgesia powerful enough to allow an incision through living tissue without the animal registering the injury. His paper was short, and the immediate response from colleagues was skepticism. The observation implied something the field had not previously considered, that the brain already contained a mechanism capable of suppressing pain completely when it was activated. It was not dulling sensation at the level of the skin or the nerve. It was something operating from inside the brain, reaching down to intercept signals before they arrived. The obvious question was what normally triggered it, and why the brain would build such a system at all. Reynolds had stimulated the periaqueductal grey. The periaqueductal grey, known by its initials PAG, is a dense cluster of neurons surrounding the cerebral aqueduct in the midbrain. The cerebral aqueduct is the channel through which cerebrospinal fluid flows between the brain's ventricles, and the PAG wraps around it in a cylinder of grey matter. Its anatomical position places it at a juncture. Above it are the forebrain structures involved in emotional processing and threat detection. Below it are the brainstem nuclei that control basic survival functions. The PAG is not a sensory structure. It processes context. What the PAG receives is as revealing as what it projects. It receives input from the hypothalamus, from the amygdala, from regions of the cortex involved in pain and fear. These are structures that process threat and emotional context, not structures that relay raw sensory data from the body. The PAG is not downstream of the injury. It is downstream of the brain's assessment of the situation. This means the descending inhibitory system can be activated not by tissue damage itself, but by the brain's interpretation that threat is present and that the body must keep functioning regardless of what is happening to it. When the PAG is activated, it sends excitatory signals to a structure in the lower brainstem called the nucleus raphe magnus. The nucleus raphe magnus, or NRM, sits along the midline of the brainstem and is a primary source of serotonin in the descending circuits. Activation of the NRM sends axons downward through the dorsolateral fasciculus, a column of nerve fibers running along the outer edge of the spinal cord. The descending inhibitory pathway draws from more than one source as well. The locus coeruleus, a nucleus in the upper brainstem that is the brain's primary source of norepinephrine, also contributes descending fibers to the dorsal horn. When the system is engaged, the dorsal horn receives both serotonin and norepinephrine from above, activating different types of inhibitory interneurons, but converging on the same functional result. Those fibers terminate in the dorsal horn, the region of the spinal cord where incoming pain signals from the body first arrive. Pain signals from the skin, the muscles and the organs travel through primary afferent neurons that synapse in the dorsal horn. From there, they would normally be passed upward through projection neurons toward the brain. The descending fibers intercept this relay. They activate local inhibitory interneurons that then suppress the neurons responsible for passing the pain signal upward. The ascending message is dampened before it can complete its journey. Opioid peptides operate within this circuit through a more direct mechanism. Enkephalins released in the dorsal horn bind to opioid receptors on the primary afferent terminal, the part of the pain signaling neuron that is about to release its signal into the synapse. The binding triggers a response through G proteins inside the cell. Voltage-gated calcium channels on the terminal are closed. These channels normally open when an action potential arrives, allowing calcium to flood in and trigger neurotransmitter release. When they are closed, less calcium enters and less neurotransmitter is released. The signal is dimmed before it crosses the synapse. A second effect operates at the receiving end. Opioid receptor activation opens potassium channels in the membrane of the downstream neuron. Potassium ions flow out of the cell, carrying their positive charge with them. The interior of the cell becomes more negatively charged. This hyperpolarization raises the threshold at which the neuron will fire. A signal that might otherwise have propagated upward now fails to reach that threshold. The neuron goes quiet. The pain that was about to become conscious does not. The connection between this pathway and the opioid system was confirmed when researchers administered naloxone during PAG stimulation. Naloxone binds to opioid receptors without activating them, blocking any opioid peptide from doing so. When naloxone was given to animals whose pain was being suppressed through PAG stimulation, the analgesia was reduced. The opioid peptides were not incidental to the mechanism. They were part of how the descending pathway was suppressing pain at the dorsal horn. The reason for this architecture is legible from its function. Pain during a genuine threat impairs the exact behavior survival requires. A person who cannot move after an injury, not because the injury prevents movement but because pain does, is at a greater disadvantage than one who can suppress that pain long enough to act. The descending inhibitory system exists to give the body a window in which to keep functioning despite damage. The pain is not gone. The tissue is still injured, but the signal has been intercepted and the brain has bought itself time to respond to the threat before registering its full cost. The pain comes later when the body can afford it. What activates this system is not always the injury itself. The PAG can be engaged before any damage occurs, triggered by the brain's assessment that threat is present rather than by pain arriving from below. The descending pathway is already positioned and ready. The clearest accounts of what happens when it fires fully are not from laboratories. Henry Beecher was an anesthesiologist who served in the Italian campaign during the Second World War. In the field hospitals where he worked, he observed something that did not match the expected relationship between wound severity and pain. Soldiers with significant shrapnel wounds, broken bones, and penetrating injuries often reported experiencing little or no pain in the immediate aftermath of being hit. This was not temporary numbness from shock. The pain those injuries should have produced was genuinely absent. Some of the men were in good enough condition to make casual conversation and to complain about secondary discomforts, while showing no distress about wounds that would have been incapacitating in another setting. A poorly placed IV line bothered them. A compound fracture did not. When Beecher asked these soldiers whether they wanted morphine for the pain, many declined. He was careful to distinguish this from stoicism. He was not describing soldiers who were suppressing a report of pain to appear strong. He was describing soldiers who sincerely reported that they were not experiencing what their injuries should have produced. After the war, Beecher published his analysis. He compared soldiers wounded at the Anzio beachhead with civilian patients who had undergone surgical incisions producing comparable tissue damage. Among the wounded soldiers, only a minority requested morphine. Among the civilians with similar injuries from surgery, the vast majority did. The environmental difference was complete. One group had been in conditions of acute life threat immediately before injury. The other had been in a controlled medical setting with no danger present. Beecher argued that the wound was not the primary determinant of how much pain it produced. The context in which the wound was received determined how severely it was registered. This was a claim about the brain, not about the injury. The experimental investigation came through animal studies of stress-induced analgesia. Animals exposed to brief unavoidable stress showed significantly elevated pain thresholds in the period immediately afterward. The elevation was measurable, reliable, and reproducible across laboratories. It was not a behavioral artifact. It was a genuine change in nociception. The same physical stimulus produced less pain response in the stressed animal than in the control. The effect followed a recognizable temporal arc. It appeared quickly after the stressor, peaked within minutes, and declined as the stress response resolved. And it appeared across species, from rodents to primates, suggesting it was an evolved response that had been conserved across animals that faced physical threat. The neural mechanism linking acute stress to analgesia runs through the same structures that David Reynolds had identified in his surgery experiments. Acute stress activates the amygdala and the hypothalamus, structures that process threat. These structures project directly to the PAG. When they fire under conditions of perceived danger, the PAG receives that signal and the descending inhibitory pathway engages. The pain suppression is not an after effect of the stress response. It is initiated at the same moment the brain determines that a threat must be addressed. The system does not wait for pain to arrive and then suppress it. It dampens the circuit before the ascending signal has completed its journey. When naloxone was administered before the stressor, the analgesia was partially reversed. Pain thresholds were lower than in animals that had received no drug, though still elevated above the unstressed baseline. The opioid system was genuinely contributing, but the reversal was not complete. Some component of the stress-induced analgesia survived opioid receptor blockade. This finding established that stress-induced analgesia was not one response, but two occurring simultaneously. An opioid arm driven by the endorphin system and a separate non-opioid arm operating through different receptors and different intracellular mechanisms. The non-opioid component involves serotonin, norepinephrine, and endocannabinoids acting on the same descending inhibitory circuits, but through pathways that naloxone does not block. The endocannabinoid contribution is significant. These are lipid-soluble molecules that cross the blood-brain barrier readily, and whose receptors are distributed throughout the descending pain circuit. Under acute stress, endocannabinoid levels rise in the brain and spinal cord, contributing to pain suppression independently of the opioid system. The brain appears to have built redundancy into this emergency mechanism, engaging multiple suppressive pathways together when the threat is severe enough that any single failure cannot be permitted. The accounts from humans are not difficult to find. Athletes have completed competitions on broken bones without registering the injury until the adrenaline resolved. People have driven themselves to hospitals after accidents that should have been immediately incapacitating. Soldiers have continued functioning through wounds that, when treated afterward, should have prevented movement. These are not accounts of exceptional individuals. The people describing these experiences are frequently surprised by them. They are accounts of a system doing what it was designed to do, engaged at the level the situation demanded, suppressing the signal that would have halted action at the moment when action was the only available option. What the person in this state experiences is not the complete absence of sensation. The damage is there. The signal is reaching the brain. What is diminished is the component of pain that stops behavior and demands response. The urgency, the incapacity, the inability to continue. The sensory information is still being processed, but the translation of that information into halting behavior has been interrupted. The opioid system is not blocking perception. It is blocking pain's ability to prevent the body from acting. The experience of full pain comes later, often suddenly, when the suppression lifts and the situation has resolved. The same system can be triggered by something far less acute. No injury is required, no genuine threat, no physical exertion. The descending inhibitory pathway can be activated by a different kind of input entirely, the credible expectation that pain is about to diminish. The brain, given a sufficient signal that relief is coming, can activate the same endorphin system without any of the conditions that normally produce it. This has been tested experimentally with a specific drug and a specific design, and the result changed what the field understood about the relationship between expectation and the endogenous opioid system. The study that first demonstrated this came from dental surgery. John Levine and colleagues recruited patients who had just had impacted lower wisdom teeth, extracted under standard anesthesia. 3 to 4 hours after the surgery, as the anesthesia wore off and post-surgical pain set in, the patients were enrolled in a blinded test. They received an injection. Some received naloxone, the opioid receptor blocker. Others received a saline solution with no active ingredient. Neither the patient nor the person administering the injection was told which substance had been given. This design controlled for the most obvious confound. The expectation of receiving a painkiller might itself produce pain relief, but the double-blind structure ensured that expectation was held constant across both groups. Among the patients who received saline, a subset reported significant pain relief. Their pain scores dropped measurably. They had received a substance with no pharmacological activity, and yet the mechanism the brain uses to suppress pain had activated. These were the placebo responders, people in genuine post-surgical pain who experienced genuine reduction of that pain after an injection of salt water. The relief was not marginal. For those who responded, it was substantial and sustained across the measurement period. Levine's critical finding was what happened when those responders were given naloxone. Their pain returned. The opioid receptor blocker reversed the placebo analgesia. Something had been activating opioid receptors in the placebo group, and naloxone exposed this by occupying those receptors and preventing anything else from binding to them. What had been activating the opioid receptors was not a drug. It was the brain's own endorphin system, triggered not by any external compound, but by the expectation that relief was coming. The design was clever precisely because of what it could establish. Levine was not simply testing whether placebos worked. He was testing whether a specific mechanism was causally involved. By giving naloxone to people who had already responded to placebo, he could ask directly, is the opioid system causally involved, or is it a bystander? The answer was unambiguous. Remove the opioid system, and the placebo effect disappears. The endorphin system was not a correlate of the relief. It was the mechanism producing it. The variability in who responded and who did not was itself informative. Among the saline group, a proportion showed significant relief and a proportion showed none. This pattern is consistent with what is known about individual variation in the opioid system. Opioid receptor density, baseline endorphin tone, and genetic variants affecting receptor function all differ between individuals. A placebo can only activate a system that is functional and available. If the system's capacity varies, the range of placebo responses should vary accordingly. The existence of non-responders does not undermine the mechanism. It reflects the real variation in the system the mechanism depends on. The 1978 Levine paper changed the standing of the placebo effect in clinical research. Before it, the placebo response in pain trials was understood primarily as a statistical artifact. baseline improvement to be accounted for rather than explained. After it, placebo analgesia had a mechanism. Mechanisms are things that can be studied, quantified, and engaged deliberately. Subsequent imaging work confirmed the finding at the level of receptor occupancy. PET studies showed measurable changes in opioid receptor availability during placebo analgesia, consistent with the release of endogenous opioids into the descending pain circuit. The saline injection had been pharmacologically inert. The state the brain entered in response to it was not. The pathway through which expectation reaches the descending inhibitory system runs through the cortex and the limbic system. The prefrontal cortex and the anterior cingulate cortex process expectation, belief, and the prediction of outcomes. These regions project to the amygdala and hypothalamus, which project to the PAG. When the brain constructs a credible prediction that pain is about to decrease, that prediction propagates downward through these connections until it reaches the structures that release opioid peptides into the pain circuit. Expectation is not in a separate category from the brain's physiology. It is a physiological state with measurable downstream consequences at the level of neurotransmitter release and receptor binding. The placebo finding also forced a cleaner account of what pain actually is. Pain is not a direct readout of tissue damage. If it were, placebo analgesia would be impossible. The damage would be there. The signals would be firing. Nothing from the brain's expectations would be able to diminish what the tissue was physically transmitting. Pain is the brain's interpretation of those signals, filtered through its assessment of context and expectation about what is about to happen. The tissue damage is real. The signal it sends is real. But whether that signal becomes conscious suffering at a given intensity is determined partly by factors that originate inside the brain. This is not a claim that pain is imaginary. It is a description of the mechanism by which the same tissue damage produces different levels of suffering in different contexts. The implication for clinical context is not peripheral. The confidence a practitioner projects, the environment in which treatment occurs, the patient's beliefs about the intervention, all of these influence the degree to which the brain's descending inhibitory system engages. A clinician who instills confidence is not merely making the patient feel better in a vague emotional sense. At the level of opioid receptor activation and enkephalin release in the dorsal horn, they are modifying the pharmacology. This is what makes the practitioner and the setting genuinely part of the mechanism, rather than decoration around it. In pain specifically, the expectation of relief is pharmacologically active. Another common claim about endorphins and subjective experience deserves the same scrutiny the placebo story received. It concerns a different context and a phenomenon that millions of people have described in their own experience. It is usually called the runner's high. The explanation people have been given for it names endorphins as the cause. At the level of the mechanism that explanation usually implies, it is incorrect. But the full account is more interesting than a simple correction. Exercise triggers endorphin release. Endorphins flood the brain. The runner's high is an endorphin rush. This explanation is so routinely offered and so widely accepted that most people assume it has been directly confirmed. It has not. The reason it has not is the same problem already established when the endorphin family was first mapped: the blood-brain barrier. During exercise, the pituitary releases beta-endorphin into the bloodstream. The release is real and measurable. Blood samples taken from people during and after sustained exertion show elevated beta-endorphin concentrations that correlate with exercise intensity. The problem is that this endorphin is the kind that cannot cross from the blood into the brain. It is hydrophilic and 31 amino acids long, with no transporter waiting for it at the blood-brain barrier. It stays in the blood. The brain is not receiving it. The direct test of this came in a 2021 study. 63 participants were divided into groups and given either naloxone, an opioid receptor blocker, or a placebo before running or walking. Naloxone occupied their opioid receptors for the full duration of the exercise session, ensuring that even if endorphins were released into the bloodstream during the run, they could not bind any opioid receptor in any tissue. If the runner's high depended on the opioid system, blocking all opioid receptors should have eliminated or substantially reduced it. The runner's high persisted. Participants who received naloxone and then ran still reported euphoria, reduced anxiety, and elevated tolerance for discomfort. The experience was not blunted. The opioid blocker had successfully occupied their opioid receptors throughout the session, and the state appeared anyway. Endorphins were not the primary driver of what was being experienced. Whatever was producing the high was not working through an opioid receptor. The participants who walked under the same conditions, with or without naloxone, did not report the high. The phenomenon was real and exercise-dependent, while ruling out the opioid system as its primary mechanism. The leading candidate for what is actually producing it is a different class of molecules entirely. Endocannabinoids are lipid-soluble compounds produced by the brain and body tissues. Unlike beta-endorphin, they can cross the blood-brain barrier. During moderate to high-intensity exercise, anandamide rises measurably in the bloodstream. Anandamide, also known as AEA, is lipophilic. It dissolves through the fatty membranes of the blood-brain barrier and enters the brain. Once there, it acts on cannabinoid receptors in regions governing mood and anxiety suppression. In these regions, cannabinoid receptor activation reduces anxiety and can produce the calm engagement and mild euphoria that long-distance runners describe. Review of the available exercise literature found that 82% of studies detected anandamide increases during moderate intensity exertion. The anandamide response is intensity dependent. It rises most consistently at 70 to 85% of maximum heart rate, a level of effort demanding enough to push the cardiorespiratory system well above resting state, but sustainable over an extended period. Below that threshold, the endocannabinoid response is less reliable. Above it, the effort shifts toward anaerobic conditions, and the window of sustained anandamide elevation narrows. The anxiolytic effect that runners describe, the shedding of worry and self-consciousness after the first difficult miles, tracks closely with anandamide activity in the amygdala and prefrontal cortex rather than with opioid receptor activation. This is not simply endocannabinoids replacing endorphins as an explanation. There is a nuance. The same central neurons that release opioid peptides inside the brain during pain suppression also respond to the physiological signals that accompany sustained exercise: rising temperature, circulating stress hormones, elevated metabolic demand. The arcuate nucleus neurons and brainstem opioid neurons do not require any molecule to cross the blood-brain barrier because they are already inside the brain. PET imaging studies using radio tracers specific to opioid receptors have found evidence consistent with endogenous opioid release in the brain during sustained running. This centrally released endorphin component is real and may contribute to the runner's high alongside endocannabinoids. It is simply not the peripheral pituitary release that the popular account has always pointed to. The full account then runs as follows. The pituitary's beta-endorphin release during exercise is genuine and detectable in blood but cannot reach the brain. Endocannabinoids rise during sustained moderate to high exertion and cross the blood-brain barrier. Anandamide is the primary one, and it acts on circuits involved in mood and anxiety. Central neuronal endorphin release inside the brain, triggered by exercise-related physiological signals, likely contributes as well. The popular account named endorphins as the cause. Endorphins are part of what is happening, but the peripheral version that gets measured in blood is not the driver, and the molecule that crosses the barrier and does most of the work is one the popular account rarely mentions. The runner's high story is wrong in a specific way. It names a real molecule, describes real effects and locates the mechanism in the wrong system. What is more interesting is what the full account reveals about the endorphin system. It does not light up during exercise because exercise is pleasant. It activates because the body is under metabolic stress, because the brain interprets sustained exertion as a physiological demand that requires suppression of competing signals. And outside of exercise, it activates in a context that appears to have nothing in common with running. The signal that triggers it there arrives not from metabolic demand, but from the presence of other people. Robin Dunbar is a British evolutionary anthropologist who has spent decades studying the relationship between neocortex size and social group size in primates. Among the patterns that emerged from this work was a correlation. Species with larger neocortices maintain larger and more complex social groups, and within those groups, across many primate species, the primary mechanism for forming and maintaining bonds between individuals was grooming. One animal beside another, working through fur with careful attention. The activity that holds primate societies together at the individual level is a form of sustained, deliberate physical contact. What grooming does to the brain was confirmed in studies using opioid receptor blockers. Primates that were groomed showed signs of relaxation and reduced stress. When those same animals were given naltrexone before grooming sessions, the affiliative effects were substantially reduced. They did not seek out grooming partners as reliably, and the calming effects of the grooming session were diminished. The social bond maintenance function of grooming depended partly on endorphin release. Being touched by a trusted partner activated the opioid system, and the warmth that followed was molecular, not just behavioral. Further evidence came from studies measuring beta-endorphin in cerebrospinal fluid following social grooming. Groomed animals showed elevated opioid peptide levels consistent with central release. The same molecular system that suppresses pain under threat was activating in response to trusted social touch. The opioid system was not specialized for pain alone. It was also the system through which the brain registered a specific class of social experience as worth seeking and sustaining. Dunbar's group size research identified a constraint: the group size that primates can maintain through grooming scales with neocortex size, because maintaining a bond through grooming is time-intensive and involves only 2 individuals at once. A chimpanzee troop stabilizes at around 50 members. An animal can devote roughly 20% of its waking hours to grooming and maintains stable relationships across that number. The 20% figure is not arbitrary. It represents the approximate ceiling on time that primates allocate to any single social activity, while still managing all other survival requirements. Dunbar's calculations produced a figure of approximately 150 for human beings, based on human neocortex size. That became known as Dunbar's number, the typical upper limit on the stable social network a human brain can maintain. The problem with applying the grooming model to humans becomes clear when the arithmetic is done. To maintain 150 meaningful relationships through one-on-one grooming would require far more than 20% of waking hours. The numbers do not close. For a chimpanzee, 20% of waking hours is enough to service around 50 relationships. Humans, maintaining 3 times as many bonds at the same individual rate, would need something closer to 40% of waking hours dedicated to grooming alone. The remaining activities that survival requires would not fit. The system that scales for chimpanzees does not scale for humans. Dunbar's hypothesis was that humans solved this problem by evolving behaviors that deliver the same endorphin-releasing, bond-maintaining function as grooming, but to multiple individuals simultaneously. He proposed laughter, music, and storytelling as the primary candidates. These are activities deliverable to a group rather than just between 2 individuals, and they appear as universal features of every documented human culture. Singing with others, laughing at the same moment, participating in coordinated ritual. These are not decorative extensions of human sociality. According to the social endorphin hypothesis, they are the mechanism that allows humans to maintain social bonds at the scale that human neocortex size predicts. The direct evidence that social laughter activates the brain's opioid system came from a 2017 study by Lauri Numanmaa and colleagues at the University of Turku. 12 healthy male participants underwent 2 PET scans each, using a radio tracer specific to mu-opioid receptors. In one session, they watched comedy with friends present, all laughing together. In another, they were alone. The design controlled for extraneous variables by holding the content constant. The difference between conditions was specifically the social context of shared laughter versus solitary viewing. The contrast was measured in terms of which brain regions showed changes in mu-opioid receptor occupancy, indicating endogenous opioid release. Social laughter triggered significant opioid release in the thalamus, the caudate nucleus, and the anterior insula. These are not the regions of the descending pain circuit. They are structures involved in reward processing, social salience, and the emotional valence of experience. The opioid system was activating in reward and social circuits because of shared laughter, not because of any physical stimulus. The same molecular event that suppresses pain at the dorsal horn was occurring through the same class of receptor in the circuits that process whether an experience is worth repeating and whether the person involved is someone worth remaining near. The Numanmaa study also found that baseline mu-opioid receptor availability in the cingulate cortex and orbitofrontal cortex predicted how frequently participants reported laughing socially in their everyday lives. People whose opioid systems were more primed for social laughter engagement were people who reported more of it. The relationship ran in both directions. The system's sensitivity shaped the behavior, and the behavior activated the system. The finding also speaks to what has been conserved across evolutionary time. The same molecular mechanism that registers the bond-reinforcing reward of grooming in a chimpanzee was registering the reward of shared laughter in a human being. The opioid system is not a human novelty. It is a primate architecture that humans have learned to activate through means the other primates cannot. What this means for the things humans do together is not a small claim. The activities that all human cultures perform in groups, persisting across every documented society regardless of geography or technological complexity, are precisely those Dunbar predicted would release endorphins simultaneously. Shared meals accompanied by laughter, music performed and heard together, ceremonies with synchronized movement, athletic spectacles that move a crowd in unison. These are not entertainment in the thin sense of that word. At the level of what they do to the opioid system, they are the mechanism by which humans maintain social bonds at a scale no other primate can reach. The system that makes belonging feel like it does is the system that makes its absence feel like what it does. The opioid receptor that mediates the warmth of social connection is the same receptor that registers when connection is missing. The molecule and the receptor are the same in both conditions, and the experience of the two states, being included and being left out, are not merely emotionally different. They share neural substrate with something older and more fundamental than emotion. Naomi Eisenberger designed an experiment in which people were excluded from a game. The game was simple, a virtual ball-tossing task called cyberball, played inside an fMRI scanner. Participants were told they were joining an online study with 2 other participants in different scanner rooms, connected through a network. The setup was designed to feel like a genuine social interaction. Before the exclusion phase began, the ball was tossed among all 3 players normally. Then the other 2 stopped throwing to the participant and passed the ball only to each other. The participant had been excluded from a virtual game by people they had never met, with a digital ball on a screen. The fact that it was a computer program producing the exclusion, not real other participants, made no difference to what the brain registered. The dorsal anterior cingulate cortex was active during the exclusion period. The DACC was not new territory in pain research. It had been identified as one of the core regions involved in the affective dimension of pain, not the sensory component that tracks where a stimulus is and how intense it is, but the component that makes pain feel bad, that generates the urgency to stop whatever is causing it. The ventral portion of the anterior cingulate cortex handles the emotional regulation of pain. The dorsal portion is involved in the distress signal itself, the message that this situation is bad and requires response. Eisenberger's exclusion condition activated the dorsal portion specifically. The neural signature of social rejection matched the neural signature of acute physical pain at the level of the specific subregion of the cortex that processes pain as suffering. The implication was significant. The brain was not simply finding social rejection unpleasant in the way that frustration is unpleasant. It was activating the circuit that activates when tissue is damaged. The brain was treating exclusion as a form of injury. The DACC was doing in response to a social event exactly what it does in response to a physical one, producing the signal that says something has happened that requires urgent response, that the current situation cannot continue, that the damage needs to stop. The overlap is not accidental. For a social primate, exclusion from the group was historically life-threatening. A chimpanzee expelled from its troop faces sharply higher mortality from predation and the loss of the cooperative resources the group provides. If the signal of social exclusion were not as compelling as the signal of physical injury, the animal might fail to respond to it with the priority it requires. The physical pain system already had the property the social exclusion signal needed. It is difficult to ignore. It commands attention above other concerns, and it produces urgent motivation to change the situation. Recruiting it for social threat gave social exclusion the same neurological weight as physical damage. The system that signals tissue injury was also made to signal social injury, because the two needed to produce the same behavioral priority. The opioid system's role in this comes through mu-opioid receptor research. People carry genetic variants that affect mu-opioid receptor expression and function, and those variants predict social sensitivity. A specific variant in the mu-opioid receptor gene, known as OPRM1A118G, has received particular attention. Individuals carrying the G allele show altered receptor expression. Studies found that G allele carriers reported greater distress in social rejection situations, and greater relief from social acceptance than A allele carriers. The endorphin system does not merely create the warmth of belonging, it calibrates how much the absence of belonging hurts. The genetic architecture of the opioid receptor predicts, at least partly, how much social pain a person experiences and how much the presence of others relieves it. A 2010 study by Nathan DeWall and colleagues tested whether a common analgesic could reduce social pain as it reduces physical pain. Participants were divided into two groups. One took acetaminophen daily for 3 weeks, the other took a placebo. Each evening, participants completed a diary measure of how much hurt feeling they had experienced during the day. Over 3 weeks, the acetaminophen group reported significantly fewer hurt feelings than the placebo group. The analgesic that reduces physical pain by dampening pain signaling circuitry was also reducing the daily experience of social pain. The circuit that acetaminophen acts on is not specific to physical injury. It is sensitive to whatever the brain is routing through its pain systems, and social hurt was being routed there. The pharmacology runs in both directions. Naltrexone studies found that blocking opioid receptors reduced the warmth typically felt during social interaction. The baseline belonging that ordinary contact with others provides became flatter under opioid blockade. Separately, social rejection under naltrexone was experienced as more distressing than without it. Opioid drugs that activate mu-opioid receptors produce a warmth and sense of connection that users frequently describe in social terms, a dissolution of the social anxiety that accompanies ordinary interaction, a feeling of belonging that the drug generates chemically. This is the same receptor activated by grooming and by laughter. The drug is producing a pharmacological version of what social bonding produces. Opioid withdrawal also carries a social dimension. Users describe the experience partly as disconnection and the absence of belonging that the drug had supplied. The opioid release that accompanies social belonging functions as a baseline that regulates how tolerable its absence feels. A person with a richly activated social opioid system who has experienced consistent bonding and connection will register the absence of that activation as painful in proportion to what has been lost. Heartbreak, loneliness, the specific sensation of social rejection. These are not metaphors for pain. They involve activation of circuits that process pain. The person who describes their grief as physically painful is making a statement that is neurobiologically accurate. What is different between heartbreak and a wound is the input, not the mechanism. The mechanism is the same. The anterior cingulate cortex activating, the opioid system registering the signal, the urgency to change the situation that both physical and social pain produce. What the opioid system can produce from the inside through the activities of social bonding has a ceiling. It is real and significant, but it is bounded by what the system was built to sustain. And then there are the external opioids, the molecules that speak the same language but at a volume the internal system was never designed to handle. Morphine does not invent a new sensation. When it binds to the mu-opioid receptor, it activates a system that was already there and already in use, a system already producing the same category of experience. The warmth it generates is real warmth. The pain relief is real pain relief. The sense of belonging users describe is produced through the same receptor that social bonding uses. What morphine introduces is not a new chemistry, it introduces a quantity. The natural system releases opioids in specific amounts, in specific locations, for specific durations. When participants in Numenma's PET study laughed with friends, opioid release appeared in the thalamus and caudate nucleus. It was measurable and significant, and it lasted minutes. When the periaqueductal grey activates during acute stress and sends inhibitory signals down through the spinal cord, it does so in response to a specific threat at a specific intensity. The response ends when the threat ends. When social grooming releases endorphins in primates, the effect sustains the bond and calibrates the baseline of belonging, but within a range the system can regulate. The internal system is not cautious by design. It is bounded by what it was built to produce. Heroin crosses the blood-brain barrier faster than morphine because it is more lipophilic. In the brain it converts to morphine, which then binds mu-opioid receptors with high affinity, and at concentrations the internal system never approaches during ordinary function. The binding is not qualitatively different from what meta-enkephalin does when it binds the same receptor at the dorsal horn. The receptor runs the same downstream signaling. Potassium channels open, calcium channels close, and the neuron quiets. But the concentration arriving at those receptors is not what the system was calibrated to receive. The receptor cannot distinguish the source of the signal. It reads the occupancy and responds accordingly, whether the molecule came from the hypothalamus or from a syringe. Consider the difference between rain and a flood. Rain falls in measured amounts, reaches specific areas, and soaks into the ground at a pace the soil was built to absorb. A flood is the same water moving to the same ground, but at a volume that overwhelms every mechanism of absorption and regulation. The ground does not respond differently because the water is different. It responds differently because the quantity exceeds what the system was designed to handle. The opioid receptor system works the same way. The molecule is familiar. The receptor recognizes it. What the system was never built to handle is the concentration at which it arrives. This is why the opioid system is the most effectively hijacked target in the nervous system. The drugs do not deceive the brain about what is happening. The brain recognizes exactly what is happening. It processes signals that mean threat suppressed, pain resolved, and crisis over. It just receives those signals at an intensity the natural system was never built to produce. The experience is not alien, it is distorted recognition. Users describe opioids using the internal system's normal vocabulary: warmth, relief, and the absence of a pain that had been constant. These are not invented effects overriding the brain's experience. They are the existing outputs of the opioid system, delivered at a magnitude that exceeds what the system can generate from the inside, what laughter produces as warmth, what belonging produces as a sense of adequacy. The drug generates all of these simultaneously. It does so at a concentration the internal pharmacy cannot match. The brain has no internal reference point for this level of activation. Laughter, grooming, and exercise. None of these approach the receptor occupancy that a systemic opioid dose produces. The system has no calibration for a signal this large because the signal it was built to receive never arrived at this magnitude. The receptor system responds the way any biological regulatory system responds to persistent stimulation. It was not built to receive. It adjusts. Receptors are internalized and sensitivity decreases. The baseline shifts. What produced an effect of that magnitude at first produces a diminished one later, requiring greater amounts to reach the same state. And when the drug is withdrawn, the system's adjusted baseline means the natural release mechanisms are no longer sufficient. Laughter, bonding, and exercise can no longer reach what the brain now registers as functional opioid tone. The absence of the drug is experienced not as the absence of something pleasant, but as a deficit. The loss of a baseline the system has reorganized itself around. That is not a description of a character flaw. It is a description of a regulatory system responding exactly as regulatory systems respond when exposed to signals at a scale they were never designed to receive. The reason the drugs work so well is the same reason they are so dangerous. The mu-opioid receptor was not built with a lock that keeps out external molecules. It was built to recognize a specific molecular shape. Morphine fits that shape. Heroin fits it faster. Fentanyl fits it with an affinity that makes morphine seem restrained. The internal system produces the same signal and activates the same circuits. The drugs speak that language at a volume the internal pharmacy was never built to produce. The receptor cannot tell the difference and responds accordingly. Long before the word endorphin existed, before the poppy was cultivated or anyone extracted morphine from it, the brain was already running this system. It was running it for the same reason it has always run it, not for pleasure, but for survival. The original question was why the brain would have a receptor that morphine fits. The answer, worked out over decades of pharmacology and neuroscience, is that the receptor was never built for morphine. It was built for the brain's own molecules, the ones the brain synthesizes and releases in response to the situations it was designed to navigate: injury, threat, and social exclusion. Morphine fits because it resembles those molecules closely enough that the receptor cannot tell the difference. The receptor was there first. The plant compound found its way into a lock that evolution had already built for a different key. What the system does is not produce happiness. It manages the body's response to events that require a response. It suppresses pain when pain would prevent survival. It releases warmth when belonging is maintained, and withholds it when belonging is threatened, producing the urgency to restore it. It opens the inhibitory pathway before pain arrives, when the brain believes relief is coming. It connects grooming, laughter, and storytelling to the same molecular signal that quiets a nociceptive neuron at the dorsal horn. The system is not the brain's reward department. It is the brain's survival accounting system. It runs a continuous calculation about threat, belonging, and whether the current situation is one the body should fight through. When Li and Chung identified the larger opioid peptide from pituitary tissue in 1976, they named it by combining endogenous with morphine. The name was accurate in one direction but easy to reverse. The molecule was not named after morphine because it resembled a drug. It was named that way because the researchers needed a word, and the word for the external compound was the only available name for what the receptor recognized. The brain did not borrow from pharmacology. Pharmacology had been operating, without knowing it, with a copy of something the brain had always made. The centuries of human use of the opium poppy were centuries of accidental access to a system the body was already running for entirely different reasons. That system is running now. It calibrates how much this moment hurts. It registers whether the people around you constitute belonging or its absence. It decides whether the current situation requires an override of the pain signal, or a suppression of social anxiety, or a brief release of warmth in response to a shared laugh. It has been doing this, unobserved and unnamed, for the entire length of human history. The scientist who found it named it after a drug because that was what led them there. What they were actually looking at was something far older, the brain's method for staying alive in a world that has always included pain, threat, and the need for others. The endorphin system did not evolve for pleasure. Pleasure is what survival looks like from the inside.